Okay. So I'm going to assume that the last recording I posted was, came out okay. I don't know if anybody had a chance to look at it, but you should be able to access it if you haven't tried to in the past.
No. So maybe we'll give people a few minutes.
Sure.
Give people a few more minutes. Maybe till about 5 after.
Yep.
And we'll get started.
Makes sense. Mm-hmm.
And by the way, everything we're presenting today is at this link. If anyone wants the PowerPoint or there's some sample files today we're going to be looking at.
Cool.
Everything is there.
And we can see your screen pretty clearly, so we're set there.
Okay. And I may have lulled you into a false sense of thinking that we're going to end early, but today it's packed. There's so much material, and we have such limited time together for the class that sometimes I go a little bit overboard, and I try to cram in as much information as I can, but we'll see as we go along. So we'll start in just a couple minutes.
Great. Yep.
I have so many clocks. I don't know what time it is anymore.
It's 4 after.
4 after? Okay.
Got one more minute.
One more minute. Okay.
Yeah.
And thanks everybody for the responses to last week's questions. We do in fact have a bit of a range of baseline genetics knowledge. And so I'm going to try to balance that today. And why don't we go ahead and get started. Today's session is the Introduction to DNA and Genetics. It's deliberately high level and covers a lot of ground with the idea that it's good sometimes to hear things repeatedly to help them sink in. And so on the one hand, for some of you, today is going to purely be a review. For some of you, today might be a little bit overwhelming. But if you're overwhelmed, just kind of sit back, try to enjoy the show, and keep in mind that all of this material is going to be presented again as we go through. So today's session is Introduction to DNA and Genetics. And if you look over the next few weeks, there's a lot of kind of foundational material that we need to cover, although in each of these sections, we'll try to weave in something new in addition to the review. So, for instance, today we'll be introducing some HL7, V2, and FIRE representations of some simple DNA variants. When we talk about the central dogma of biology, we'll try to bring in some topics on epigenetics. But the foundational material in the early weeks will hopefully really set the stage for some of the more complex and interesting topics coming up in subsequent weeks. And please feel free to send feedback are we going too slow? Are we going too fast? Are we hitting it just right? Is there material that you thought you'd see that we're not seeing, etc.? Okay. So the way I like to do the class is to try to begin every session with a clinical case. And the idea is that the clinical case illustrates the topics that we're going to be covering during this morning's session. So today we're going to talk about sickle cell anemia. It's a little bit tough sometimes to know, do you present the case ahead of time before the material, or do you present the case after the material, especially early on where some of the fundamentals may not be well understood. So as we go through the case, there's going to be a couple of pieces of that case where we won't have explained yet as part of the class. But once we get into the third and fourth week, it'll be a little bit easier to just start off with the cases. The cases will introduce new material, which we'll then cover through the course of the class. So today we're going to talk about, possibly one of the most common genetic disorders, sickle cell anemia. We're going to talk a little bit about what has led to this explosion in popularity for genetics. We're going to go over some fun facts, which are really just some, I guess I might say, mind-blowing statistics to give you kind of a super high-level picture about DNA. And then we're going to dig in to probably what's going to be a review for a lot of you, but we're going to go through it pretty quickly, just basic DNA structure so that we're all on the same page. What's a chromosome? What's a gene? How do you represent a gene in a database? And then we're going to come back to the clinical case, and we're going to actually take a look at how sickle cell anemia would be represented, now that we understand it, how it would be represented using HL7 standards. And then every week there'll be a slide for vocabulary and a slide for references. We're not going to talk about the vocabulary or the references at all. We'll actually cover the vocabulary during the course of the presentation, and the vocabulary is just a summary of the key vocabulary topics that we covered. And likewise with references, we're not going to specifically look at the references, but you'll see as we go through that there's a lot of material that we addressed, and all of that material is pointed to either in the individual slides or in the references. And in fact, if you download the slide deck, you'll see, for instance, there's text corresponding to the slides in the little notes section. For those of you interested in reading a little bit more about each one of the slides. Okay. So let's start off with sickle cell anemia. And probably most of you are familiar with sickle cell anemia. And in this particular case, this is a 6-month-old girl who was, this was a 24-month-old girl who was brought into the emergency room for the second time in 6 months. And she was brought into the emergency room because she refused to bear weight on her feet. There was no history of fever, infection, or trauma, and she had an otherwise unremarkable medical history. And the last time she had been seen in the emergency room 6 months ago, they noted that her hemoglobin level was a little bit low. She was mildly anemic. They noted that her spleen was slightly enlarged. And this time, when they examined her, they also noticed that her spleen was a little bit enlarged, and her feet and her hands were swollen and tender. And in fact, she refused to stand up. When the clinicians questioned the family members, it turns out that both parents had siblings who had died in childhood of infections, and others that may have had sickle cell disease. And so based on that, they tested this particular patient and in fact determined that she had sickle cell disease, and they diagnosed her with sickle cell anemia. And what you see in this picture is on the left. This is her hand at the time that she presented to the emergency room. And then this one here is a picture of her hand 2 weeks later. And while the picture's a little bit fuzzy, I think you can see here the bone actually looking a little bit of moth-bitten. You know, you'd expect to see the borders of the bone being nice and smooth, but here they look a little bit ratty. And so how is it that a particular genetic disease can result in such striking changes to the bone and such intense pain? And that's what we're going to explore here. What I want to do for each of these cases is to introduce you to the notion of and the importance of family history. Really, you know, we'll talk a lot about genetic testing as we go through the course, but there's really no single more important test than the family history. And in fact, this is how generally you even know that a disease is genetic in the first place. And so this isn't necessarily the pedigree for the patient in question, but it's a classic pedigree that you'll see for a genetic disease that's called autosomal recessive. So this particular pedigree represents an autosomal recessive inheritance pattern. The way you read these is the square boxes are male, the circles are female. These are married couples, and then these are their offspring. And if it's got a little dot in the center, it means that this person is a carrier of the disease but doesn't actually manifest the disease. And if the circles or squares are filled in, this means they actually have the disease. And so what you see here is a man and a woman, each who are a carrier of sickle cell anemia, wind up having 5 children, 2 of which are affected by the disease. And this is, as we'll talk about when we have the session on family trees, this is a classic autosomal recessive pedigree. And again, we haven't really talked about what do we mean yet by carrier. And so I'm going to just move on. But as we go forward today and in a couple of weeks, we'll explain exactly what we mean by the fact that these people have the gene, where they have one copy of the gene, so they're carriers, but they don't manifest the disease. And so we'll be explaining that as we go on. For some of the diseases, I think it'll be interesting to pull in a smidge of population health considerations, although honestly, we're not going to be spending much, well actually, honestly, we're going to be spending very little time talking about, in this class, topics such as evolution. We're not going to be talking about topics such as population health. Really what we're going to be doing in this class is focusing on, you have a patient in front of you, they have a genetic disease. How can we understand the pathophysiology and the molecular biology of that genetic disease? And what then are the implications for an informatics perspective, for data communication, for data storage, for looking up the particular variant to determine its clinical significance? But in this case, the population health is pretty interesting because of the overlap in the geographic distribution of the sickle cell gene with the distribution of falciparum malaria. And there's a lot of diseases that overlap in their geographic distribution with malaria. And in this particular case, it's thought that the gene, the mutation that results in sickle cell disease, is actually protective against malaria. And so this particular mutation was selected for. There was a lot of selective pressure for this gene to propagate, for this mutation of this particular gene to propagate, because it helped people survive to reproductive age. And by the way, stop me along the way for questions. So here we have a slide showing normal red blood cells on the left and sickled red blood cells on the right. And probably people realize that red blood cells contain the hemoglobin protein. And what's happening here on the right in these sickle cells is that the hemoglobin protein, because that protein, there's an alteration in the protein as a result of the genetic mutation. The alteration in the hemoglobin protein, what that does is when the red blood cell is subjected to low oxygen states, so initially the blood is highly oxygenated, but then after it delivers its oxygen and it's working its way back past the veins, it's in a low oxygen state. And in a low oxygen state, these abnormal hemoglobin molecules can clump together and form these polymers. And these polymers distort the shape of the red blood cells. And as a result, these red blood cells can get trapped and stuck, and they also become fragile. And so as a result of the low oxygen state, the red blood cells sickle, they get plugged up in capillaries, they become fragile, the red blood cells get destroyed, you get anemia, and the red blood cells clog up the works, causing symptoms, pain, and in fact causing the destruction, the bony destruction that we saw in the previous slide. The hemoglobin molecule, actually the hemoglobin in red blood cells is comprised of 4 hemoglobin chains all connected together. This is one of the 4 protein molecules comprising a complete hemoglobin, and this is called the beta globin chain. This particular protein, this beta globin chain, is produced by the hemoglobin beta gene, or the HBB gene. And so what's happening here is a mutation in the gene is resulting in a change in this protein. One of the amino acids in this protein gets changed, and as a result of that change, this protein, when it's under low oxygen situations, will clump and form polymers. And the pathophysiology of the disease is kind of summarized here, where what you have is hemoglobin, when it's in an oxygenated state, is just acting like normal, but in a deoxygenated state, the hemoglobin will form these polymers, and these polymers will distort the red blood cells, and these red blood cells will then clog the works, causing the pain. We haven't yet talked about what this stuff on the left means, and I'll tell you briefly, but we will be covering this a little bit later today, and then we'll be covering this again over the next couple of weeks. And I think most of you may remember this from your high school biology class, is that the DNA has a G, then an A, then a G, undergoes a mutation where this A, it becomes a T. And as a result of that, within the beta globin protein, glutamic acid amino acid is replaced, and now you have a valine amino acid. And the valine amino acid, if you will, is a little bit more sticky than the glutamic acid amino acid. And because of the stickiness of the valine amino acid, the hemoglobin molecule is prone to clumping, particularly under deoxygenated states. So again, hemoglobin, just to summarize, sickle cell anemia, you have a point mutation in the DNA. That point mutation in the DNA results in a corresponding amino acid change in the protein. As a result, that protein, when it's under deoxygenated conditions, can clump. That clumping leads to distortion of the red blood cell, and that distortion leads to vaso-occlusion with pain symptoms, but it also leads to greatly shortened red blood cell lifespan, and that's what leads to the significant anemia. Okay. All right. Any questions before I go on? Okay. So let's now shift topics, and we're going to talk about why genetics has become so popular. Genetics has always been a big deal to a lot of people, but it seems like lately you can't go anywhere, you can't read anything, you can't pick up a magazine without finding something about genetics or CRISPR. And I believe that the tremendous growth in popularity are largely because of these two factors. One having to do with sequencing and the fact that we're just sequencing mountains of DNA in ways we never could before. And number two has to do with CRISPR-Cas9 or genetic engineering capabilities, with the notion that not only can we now sequence a boatload of DNA, which allows us to see and learn what the genetic basis is for a lot of different disorders, but with CRISPR-Cas9 and the explosion in genetic engineering technology, we now appear to be close to having the capabilities of actually doing something about it. So we can learn about it, what the heck is going on, what's the genetic cause, and now the prospects appear to be very good for doing something about it. So this is from, and by the way, again, for all of these slides, the sources for all of these graphics are all down in the notes if you want to see any of this stuff in more detail. But this is from the NCBI, and it shows a couple of things. One is the growth in the number of bases that are stored in their repository and the number of sequences that are stored. And you can see it's just, there's no end in sight. There's no end in sight. Possibly for GenBank, there's a little bit of a plateau, but for whole genome sequencing, these red lines here, it doesn't appear to be anywhere near a plateau to the exponential growth in data volume being captured in the GenBank, in the NCBI data repository. So we're sequencing like crazy. And also what we're seeing is a tremendous decline of DNA sequencing costs. And up until about 2000, probably late 2007, we were seeing a decline in sequencing costs, which kind of correlated with Moore's law, which is every 2 years the cost would cut in half. So here we were, and this line here shows Moore's law, which is about a 50% reduction in cost every 2 years. But then in early 2008, you see this dramatic dropping in costs. And around early 2008, really what we're seeing there is the time when the sequencing centers transitioned from the old school sequencing methods, the so-called Sanger-based methods that we'll talk about, to the second generation or next generation sequencing methods. And so this inflection point here really reflects the introduction of next generation sequencing. And you can see what a hugely dramatic change it's had, and even this slide only goes up to 2015. And I mean, it was only, I think, a week or so ago that Illumina announced they're on the verge of hitting the $100 genome mark. And so it looks as though it's not going to be too long before you can do a complete genome sequencing for just $100. Right now, it's still on the order of roughly $1,000 to $2,000 if you want to do somebody's complete genome sequencing. So these tremendous decline in costs then, of course, leads to this tremendous growth in data. And so this then leads to this tremendous interest in genomics, because now we have all this data that we can study. And in fact, look at this, you can buy your own sequencing system. You can just go buy a little sequencer from Illumina or from some other company And I just thought it would be kind of fun to pull in this picture of the advertisement. You want to do your own DNA sequencing, just, you can go buy your own little machine here now and just do it. But along with our ability to sequence DNA and, begin to make some inroads into, understanding the genetic basis of life itself or various diseases, has also, the other thing that's been going on is our ability to do something about it. And probably, I'm guessing most of you have at least heard of CRISPR. Is there anyone on the call that's never heard of CRISPR before? Or is not embarrassed to say that they've never heard of CRISPR before?
I never heard about CRISPR.
Okay. Thank you for being brave, Rafiq. Yeah. So CRISPR is a technology. We'll talk about it during the genetic engineering session. CRISPR is the hot new technology that lets us actually go in there and edit DNA. So theoretically, for instance, I could take some stem cells from you. If you have a disease like sickle cell disease, I could remove some of your bone marrow stem cells. I could actually edit them with CRISPR to fix your genetic mutations. I could then take those bone marrow stem cells and I could squirt them back into you, thereby literally curing you of your genetic disease. So CRISPR has really caught the public attention because of its potential. And I'll say potential, and when we get into genetic engineering, we'll be a little bit more sobering in what CRISPR actually does and what its limitations are and where things are going with CRISPR. But CRISPR really caught the public's imagination with the prospects of editing the human genome and curing disease and allowing us to, explore life, if you will. And what this slide represents is, this is just a simple PubMed query for CRISPR, and you can see how many articles were indexed in PubMed every year. And so there's been a doubling every year, and here we are, not even halfway through the month, not even halfway through 2017, and we're already up to 1,100 articles on CRISPR. So there's a tremendous interest in CRISPR and tremendous number of publications. And not only that, there are now people selling do-it-yourself bacterial CRISPR kits. And there's high school students that are playing around with CRISPR as part of their high school science projects. And people are playing around with CRISPR in their garages. They're mucking with plants or they're mucking with, whatever. So between the drop in cost of sequencing, the enormous increase in volume of sequencing data, and the ability to edit DNA using something like CRISPR, I think those have contributed to this tremendous increase in interest in genetics. When I put this fun facts slide together, I'm just going to check on time here real quick. Okay, when I put this fun facts slide together, it dawned on me afterwards that some of these facts aren't really fun. Some of them are actually kind of tragic. But anyhow, I call this slide fun facts only because some of these statistics kind of like spark the imagination and make us all take a step back and wonder, ponder a little bit about what the heck is going on. And so let's go through these. The human genome, if you look at, so we have 46 chromosomes in each cell, and so you have two sets of 23 chromosomes. And if you look at those 23 chromosomes, what you'll find is that there's 3.3 billion base pairs. So that's a lot of base pairs in every cell. And each cell contains about 6 feet of DNA. So if you take all those chromosomes and you unravel the DNA and you stretch it out, you actually wind up with about 6 feet of DNA in every single cell. And when you consider that we have about 100 trillion cells in our body, then all the DNA in one person's body laid end to end would be 100 billion miles. I mean, 100 billion miles? It's like, I mean, I can't tell you how many times I've gone over this fun fact and said, am I doing the calculation wrong? We have literally 100 billion miles worth of DNA in our body. So you're welcome to do the math yourself, but that's what I keep coming up with. Within our DNA, we have about 25,000 protein coding genes, which is only 1.5% of our DNA. The other 98.5% of the DNA is other stuff. It's not used to create proteins. It's doing a lot of other stuff, and we'll talk about that, the significance of the other stuff as we go through it. But I'll tell you now that for a lot of the other stuff, well, it used to be given this label of junk DNA. People used to say, well, 1.5% of our DNA is important for encoding proteins. The rest of it is junk. It just serves some kind of junk carryover evolutionary process. And we know for a fact now that that's not true. We know that the other portions of our DNA serve critical regulatory roles, but we certainly don't know the role of huge amounts of our DNA. So of the 98.5% that is not protein coding, we are making inroads into understanding some of the regulatory roles that it plays, and we'll talk about some of that, but I'll also tell you that there's huge stretches of DNA for which we don't know what its purpose is. The difference between two random individuals' DNA is 0.5%. So if I took your DNA and I took my DNA, we'd find that 99.5% of the bases would be identical, but 0.5 would be different. And when you're talking about 3.3 billion base pairs, or really 6.6 billion base pairs, when you're considering that half your DNA came from mom and half your DNA came from dad, 0.5% of a humongously big number is still a big number. So the differences between two random individuals still is up there in the millions, even though it's a small percent. When a cell divides, the DNA has to be replicated, okay, so that when the cell divides, you have a copy of the DNA in each of the daughter cells. And the process is pretty good. It's pretty accurate, and there's a proofreading correction mechanism in the cell. But even so, after the cell divides, there are still, on average, 0.7 mutations per cell division. So you look at 10 cell divisions, and there are 7 mutations that have been introduced just because of the process of replication. To me, that's a bit of an astounding number. So every 10 cell divisions, 7 new mutations are introduced. And, when we think about cancer, that begins to help us understand, because all cancers are due to genetic mutations. They're due to successive genetic mutations. And so even though the process used is very precise, we're talking about 6 billion base pairs in a cell, and so 0.7 and we're talking about millions and millions of mitoses and cell divisions that are occurring throughout the life of an individual. So we know right now of at least 5,000 genes in the human genome that are known to be associated with a disease or a trait. a newborn has approximately 75 new mutations not present in either parent. And these are from mutations that either arose during the process of sperm production or that arose during the process of egg production. So these are mutations. Mom didn't have the mutation, dad didn't have the mutation, but when the sperm was produced during that final mitosis or during that final meiosis, a new mutation during that process was introduced. So every newborn has about 75 new mutations, but you figure, 98.5% of our DNA is not directly responsible for protein coding genes. So the vast majority of these mutations are just silent. They're most of the time of no clinical significance. 1 in 100 persons is likely to receive a new mutation in a known disease-associated gene. Okay, so maybe 1 in 100, but even then you have some redundancy because you get a copy of the gene from mom and you get a copy of the gene from dad. So even though 1 in 100 persons may get a new mutation in a known disease-associated gene, that doesn't necessarily mean that it's clinically significant, and that will make a lot more sense as we go through as well. Every person has about 250 to 300 genes with likely loss of function variants, and about 25 genes predicted to be completely inactivated. So, one way of saying that is we're all mutants in a way, right? Each of us are mutants, and we all have genes that are inactivated. The complete inactivation may only be on one copy. So you might have a mutated copy and you might have a normal copy, and as a result of that, you might have no obvious manifestation, or you might have a subtle manifestation that reveals itself somehow in your phenotype. 4% of the population carry the cystic fibrosis gene, and 7% of California African American newborns carry the sickle cell gene mutation. So 1 in 14. So sickle cell is very common, 1 in 14, and cystic fibrosis is also very common. 4% of the population carry the mutation. Now, as we'll talk about in a little bit, the fact that you carry the mutation, it just means that you have it in the chromosome that you inherited just from mom, or in the chromosome that you inherited just from dad. And you don't actually manifest the disease until you have the mutation in both sets of genes. So 4% of the population are carriers for cystic fibrosis, but that's not the same as saying what the actual disease prevalence is. Okay. So let's see. Any questions at this point?
What's the ratio between carrier and actual disease? Convert that carrier into disease.
It's roughly you square the carrier frequency, because if you assume that dad has a probability of having the carrier state of 4%, and some random woman he meets has a 4% probability of being a carrier, then 25% of their offspring would actually have the disease, and so you could calculate the frequency that way. And we'll cover this in a little bit more detail when we talk about the autosomal recessive inheritance pattern in a couple of weeks.
Okay, great.
Okay, so sobering facts, but also, pretty interesting facts when you see that your DNA doesn't just sit there and faithfully carry its way through all the way throughout your life. It's undergoing a lot of mutations here and there. So DNA structure, everyone who responded to the questions knows that a cell has a nucleus, and the nucleus contains chromosomes, and the chromosomes are comprised of DNA. And sometimes you'll see a picture of a chromosome just as a linear string. And sometimes you'll see a picture of a chromosome as an X, okay, as a double string. And what the X represents is a chromosome that's been replicated and it's ready to split during an imminent mitosis or meiosis. So normally the chromosome exists in the cell just as a single string, but when the cell is getting ready to divide, the DNA replicates. You now have two copies, really, of that chromosome. They're held together at this little point here called the centromere. And now these are replicated chromosomes. But in pictures you see of chromosomes, sometimes you'll see these linear strings and sometimes you'll see the X's. And the X's are actually, they show up better in pictures, and that's why you'll often see chromosomes as an X, even though honestly, you know, the vast majority of the time, chromosomes really exist as just stretched out strings. You can take a cell and you can grab the chromosomes and look at them under a microscope, and that's what we see here. These are the chromosomes from a single cell. And these chromosomes, let me just, okay, this is the chromosome here from mom, and this is the chromosome here from dad. Each of these actually is a small X, but the X has been so collapsed that this is really an X from mom where the arms are so close you can't see them. And this is an X from dad, but the arms are so close you can't see them. And that's probably a subtle point. You're wondering why I'm bothering to raise it. But these chromosomes here actually are in this form. They're not in this form. Most of the time in the cell, chromosomes live as extremely elongated, stretched out structures. One chromosome may occupy a large portion of the nucleus. It's only when the cells are getting ready to divide that the chromosomes condense. They condense and replicate, and it's only when the chromosomes condense that they're able to be visualized like this under a microscope. So the fact is, these are condensed chromosomes that have been replicated, and therefore they're visible when they're stained. And what you see here, for instance, this is the chromosome number 1 that came from mom, and this is chromosome number 1 that came from dad. We have 22 chromosomes, 22 paired chromosomes, plus we have an X chromosome. Men have an X chromosome and a Y chromosome. Women would have 2 X chromosomes. The chromosome numberings are based just on size. And so the only reason that this is chromosome 1, 2, 3 is just because 1 was the biggest chromosome. In this chromosome, you have various genes. So in this region of the chromosome, there might be a number of genes, and in the corresponding region of the chromosome you got from dad, there might be a number of genes. But let's say, for instance, that there's a gene here, and then there's the corresponding gene here, okay? Now, the gene that I got from mom might not be exactly the same as the gene I got from dad. For instance, in the hemoglobin gene, I may have gotten a hemoglobin gene from mom that had the sickle cell mutation, whereas the hemoglobin gene I got from dad may not. So when we talk about genes, you'll often hear the word called allele, A-L-L-E-L-E, allele. And what allele represents is the particular form of that gene. So there might be the hemoglobin gene that has the sickle cell allele, and there might be the hemoglobin gene that's normal. So we'll talk about alleles quite a bit as we go through, but you can have the allele you get from mom can be the same as the allele you get from dad, or the allele you get from mom can be different than the allele you get from dad. If the allele you get from mom and the allele you get from dad are the same, what do you call that? Who's out there? I need to hear you guys. Can you guys hear me?
Yep, we can hear you.
We can.
Yeah, okay. So when the allele you get from mom and the allele you get from dad are identical, what do you call that state? That would be called the homozygous state, right? That would mean that this particular person is homozygous. They have a homozygous state. That means the allele that they got from mom is identical to the allele they got from dad. They have the exact same allele on each chromosome. What if the allele they got from mom is different from the allele they got from dad? What would you call that?
Heterozygous.
There you go. All right. Very good. Okay. So if the alleles are the same, then the patient is said to be homozygous at that particular gene or that particular site or that particular locus. If the alleles differ, then the person is said to be heterozygous at that particular gene or that particular locus. It used to be that the term allele was only applied to genes, but now the term allele is also being applied to regions of DNA in between genes. So I might be looking at a long stretch of DNA between two genes, and I might say, oh, on you have this particular string of bases, but on me, I have a slightly different string. That means you and I have different alleles. The other thing that gets confusing is in an individual person at this particular gene, they can either have the same or different alleles, but an individual person can't possibly have more than 2 alleles at this site. But in the population, there might be many different types of alleles. So if you look at the hemoglobin gene, this person might have a normal allele in one copy, and they might have the sickle cell allele in another copy, but some other person in that same gene might have yet another allele that produces yet another disease such as thalassemia. So there might be many different alleles or many different variants of a gene or many different alleles or many different variants of a locus in between genes. An individual person will either be homozygous or heterozygous for that allele, but within the population, there might be multiple alleles. So we're going to come back to this because it's so important. But before I move on, is there any questions about that? Okay. So I want to hear you guys talk a little bit. So there's an error on this slide. Okay, and I want to just, when somebody finds the error, shout it out. Probably most people are familiar with DNA, how it's two strands that are connected by these bases. A couple things in particular to point out is that the strands of DNA are anti-parallel, meaning this strand goes from the 5' end of the molecule to the 3' end, whereas this particular strand goes from the 3' end to the 5' end. Along the way, along this backbone are these bases, okay? And you can see here the color key for what bases there are. Base pairing is always complementary. So whenever one strand has an A for adenine, the complementary strand is always going to be a T for thymine. Likewise here, if one strand has the cytosine for a C, the complementary strand is always going to have a guanine. So a strand of DNA will have one of 4 bases sticking out from it at every site, and then the anti-parallel strand of DNA will be connected to it using the complementary base. What's nice about this is it lends itself to a very simple representation that's shown down here. We can represent the 5' strand of DNA just by single letter codes corresponding to the bases. So this strand here can be represented with an A, here's for base adenine, a G, here for base guanine, a T, here for base thymine, an A, here for adenine, and again, the whole strand of DNA can be represented just as a series of letters. And the complementary strand can be represented just as the complementary set of letters. Okay? Anyone see the error in the slide?
GC are parallel.
GC
second,
parallel.
third is strand,
Sorry?
right? Yeah.
Wait, say again, I'm not following you.
So on the third strand, you have GC is same parallel. It should be.
Here? Here,
Yeah.
right? And what's the error?
It should be opposite, right?
There's a
What's that?
GG bond.
Here, right?
G should be, yeah, GG and GCG. So it should be GCG, right?
This should be a C.
Yeah.
Right. Right. Because this violates the complementary base pairing rule. Okay. So really, when we talk about DNA, I mean, 90% of the class when we talk about DNA, we're not going to be looking at pictures anymore. We're going to be talking about this string of bases. And we're almost always going to be looking at the 5' to 3' strand. We're not going to be looking at the 3' to 5' strand, but that's mainly because the genes are oriented 5' to 3', and this is the way that the DNA is read. And I'll show you this in a subsequent graphic, but it's the 5' to 3' strand that corresponds to the orientation of the gene that gets turned into a protein. So we'll almost always be looking at the 5' to 3' strand. Any questions about how this picture kind of jives with this representation? Okay. So here's the DNA, but actually the way that the DNA exists inside the cell is that the DNA is heavily wrapped with proteins, and it's tightly integrated with proteins, and it's coiled, and then it's supercoiled, and then it's supercoiled yet again, So we see the DNA here being wrapped around these histone proteins. And so we have a cluster of 8 histone proteins here with two strands of DNA wrap around it. Then you have another one of these blocks, and each one of these little blocks of DNA wrapped around a group of histones is called a nucleosome. And then all of these nucleosomes wrap even tighter into these solenoids. And then all of these coil. And these coils are actually pretty interesting because the coils appear to provide three-dimensional mechanisms by which gene expression can be regulated. So the coils can block access to DNA regulation. The coils can also open up to enable regulation of gene expression. And when we come back to talking about the central dogma of biology, and when we talk about epigenetics, one of the things we'll talk about is, okay, so here's this DNA, and this DNA possibly has a gene, but, you know, not every gene of every chromosome is expressed in every cell of the body. You know, some genes are only expressed in certain cells, and some genes are only expressed at certain times. And, you know, what the heck is contributing to that regulation? And it partly has to do with the superstructure of the DNA, what portions of the DNA are exposed, and it also has to do with the histone proteins and that type of regulation. Okay. A little bit more in depth on the DNA structure. So, because I want to also emphasize a couple of things. One is this notion that one strand is 5' to 3', and the other strand, so here's the 5' to 3' strand, and then here's the anti-parallel 3' to 5' strand. And it'll make more sense as we go along, but you have one strand here, and then this strand has a series of bases sticking out from the sugar backbone, sugar phosphate backbone, and then these bases have hydrogen bond connections to the complementary bases on the anti-parallel strand. And that's what comprises the DNA. Um, 50 years ago, when we thought about chromosomes, this is the way we thought about chromosomes. We thought about a chromosome having genes, and we thought about there being junk DNA in between the genes. Right? So you had a chromosome with some genes and a bunch of junk DNA. Then we started to learn more. Also, it's interesting to ponder for a minute. Okay, so here's the chromosome. It's a string of bases, and there's nothing structural that demarcates the gene. Okay, so how does the cell know that this is where a gene begins? Okay, so all this is a string of bases. There's nothing structural, there's nothing physical that demarcates, hey, guess what, cell? A gene starts here. So how does the cell know that this is where a gene begins? Okay, so hold that thought. Hold that thought. Let's come back to that in a minute. But for now, I just want to kind of get you thinking a little bit about how does the cell know where a gene begins? Okay. If you look at human chromosome 11, and so this is a graphical representation of chromosome 11. Human chromosome 11 is about 135 million bases. You'll see this notation MB. quite a bit. The capital M stands for mega or million, and the B stands for bases or base pairs. So this would be a stretch of about 135 million bases. This is a fairly large chromosome. It's about 4.5% of our DNA, and chromosome 11 contains approximately 1,350 genes. Notice that everything is approximate also. Notice that I'm not telling you exactly how many protein coding genes are on here, because as we'll talk about in a little bit more detail, we don't exactly know where all the genes are on all the chromosomes. You know, there's no structural mark that says, here's a gene. Sometimes we have to kind of guess, well, we believe this must be a gene because, well, we found a messenger RNA or we found a protein, and there must have been a gene responsible for this messenger RNA, or there must have been a gene responsible for this protein, and we can infer what the DNA sequence must have been in order to yield this particular RNA or this particular protein. So, there's probably a gene right here. We have no idea what it does, but you know, there's probably a gene that's probably right here. If you look around on chromosome 11, just like any other chromosome, you'll find that the genes are not randomly distributed. The distribution is not uniform. What you'll find is that there's gene-rich and there's gene-poor areas. And really, in this class, you know, I'm not going to so much explain the why. I mean, I don't know that I could explain the why. I'm going to simply try to say, you know, this is the way it is. So we believe that there are gene-rich and gene-poor areas on our chromosomes, as illustrated by this slide. And on average, a chromosome has about 6.7 genes per million bases. Chromosome 11 is a little bit richer in its genes. It has about 10 genes per million bases, but there's also gene-poor chromosomes like chromosome 4 or chromosome 13. And why that is, I'm not going to conjecture here, but it is what it is. Since the late 70s, we realized that our notion of a chromosome from the 50s could be expanded. We learned that there are stretches that the genes are split or interrupted, if you will, by what were termed introns. So here's the gene, and the gene is really comprised of exons, which ultimately these three exons will get connected together and the introns will get removed during the RNA processing and the protein translation. So a gene is really comprised of exons and these intervening introns, and only the exons are used in the downstream creation of proteins. The introns aren't. We also learned that adjacent to the start of a gene are these regions we call promoter regions. They're just slightly upstream from the genes. And these promoter regions bind to factors such as DNA or RNA polymerase, and that's one of the mechanisms by which the body knows, hey, here's where the gene begins. I didn't necessarily know that a gene begins here, but there's a sequence of DNA here that has a particular, what's called a motif. The promoter region has patterns of DNA recognized by RNA polymerase. The RNA polymerase knows, recognizes them, binds here, and then initiates transcription. And so we discovered that there are these promoter sites slightly upstream of genes, and that these promoter sites initiate transcription of the gene. And here's another picture illustrating that. This is the gene responsible for sickle cell disease. This is the hemoglobin gene. And again, hemoglobin is a little bit complicated because the hemoglobin molecule is actually comprised of proteins from the beta globin gene, but it's also comprised of proteins from a separate gene called the alpha globin gene. So the mutation in sickle cell disease is a mutation in the beta globin gene. The beta globin gene encodes 2 of the 4 components that comprise a mature hemoglobin molecule. What we see in genes is exons and introns. We see that there is a promoter region just upstream from the gene, and that's what signals the RNA polymerase to begin transcribing the gene. Okay. We see that the gene is comprised of exons and introns. The exons, the first exon, is where transcription begins, and it's a little bit difficult to show this picture and explain it until we've talked about the central dogma of biology, so we're going to be revisiting this picture at that point. But let me say here that the exons are ultimately what are transcribed into messenger RNA, but not all of the messenger RNA ultimately is translated into protein. For the first exon, even though all of that exon is turned into messenger RNA, the actual protein translation starts partway into that first exon, and protein translation ends partway through the last exon. And if you look at the beta globin gene, for instance, you'll see, okay, it has 3 exons. Transcription starts with the first exon, and translation into protein starts right where it turns green, and translation into protein ends right here where the green stops. And upstream from the beta globin gene, we see what are called motifs, or regions of DNA, patterns of DNA, or short sequences of DNA that we find upstream from a lot of genes, and we've come to recognize those as DNA polymerase binding sites. And so here upstream from the beta globin gene, we find what's called the CAT box and the CATA box. You'll see a short sequence of DNA slightly upstream from the start of the beta globin gene that has the CAT motif, and you'll see a short segment of DNA slightly upstream that has the TATA motif, and we recognize those now as RNA polymerase binding sites. And it's significant because you can have mutations in the promoter region and not have any mutations in the gene, and just having mutations in the promoter region might result in lack of binding of RNA polymerase, and might result in, therefore, this gene not being transcribed. And so all of a sudden you have no hemoglobin, even though when you look at the hemoglobin gene, you don't see any mutations in it whatsoever. People have probably heard of the BRCA1 gene that is, when it's mutated, can result in breast cancer. The BRCA1 gene has 24 exons, and you can see almost all of this gene is introns. And then you have this other gene here, the beta myosin heavy chain gene, and this has 40 exons. So these are some big genes. Some genes are very selective in the cells in which they are expressed, whereas some genes are more constitutive. They're expressed in almost every cell. So beta globin, for instance, is highly selective. You know, in red blood cell precursors, you have tons of hemoglobin being produced, but in liver cells, for instance, you have no hemoglobin being produced. And in these very selective cells, in the genes that are very selective, you'll often find the cat and the tatabox promoter regions, whereas in the constitutively active genes like BRCA1 gene is responsible for DNA repair. And so every single cell expresses BRCA1. Oftentimes the upstream promoters that you see are less well understood. You won't necessarily find the classic promoter motifs, but instead what you'll find upstream are areas very rich in CG pairs. So C for cytosine, G for guanine. So upstream, for instance, of the BRCA1 gene, you'll find that there are a number of CG pairs, or what are called CG dinucleotides. And we'll come back to that, the importance of that, when we talk about epigenetics during our central dogma discussion. Okay, any questions before I move on? Or you know, in my mind, I look at this and it's like questions, you know, what can you do other than just accept this and kind of be marveled by how all this works? And you know, you either have a million questions or you just accept it, you know, but anything else on this one before we move on? You know, it is what it is. And, you know, some people have asked me before, you know, how do we know all of this stuff? And, you know, you know, some of this stuff is really just, yeah.
I'm sorry, I had myself on mute and was asking you a question earlier. But I was wondering, as we get further into the course, if you might be, or if you're going to be talking about, you know, basically where clinical interventions might take place with respect to, you know, promoter regions, introns, exons. You know, you talked a little bit about, you know, gene correction, but, you know, some examples of those as we get further into the course.
Well, so we have, I think it's week, let me see, it's on July 6th, we have an entire session devoted to genetic engineering, and the whole focus of that session is going to be on clinical interventions.
Okay.
So,
yeah.
You know, let me know, Jim. We have a whole session. You know, I thought about trying to also introduce some of the topics along the way, which maybe would be helpful. So right now it's primarily concentrated during the genetic engineering session without a lot of interventions along the way. And partly that's because the field is so new. I was looking at clinicaltrials.gov just a couple of days ago for, you know, sickle cell trials, and they certainly are going on out there. And maybe towards the end of today we can even talk about where we, you know, what some of the trials are for sickle cell disease. Sure.
Yeah, and that makes sense. And you know, I was just thinking that it might be nice, you know, kind of going through you know, as we get into some of these areas, to kind of stub out, you know, discussions for future sessions to say, hey, like for example, if this was a gene sequence that was related to cancer, there might be not only gene therapy but modifier therapy that might interfere with promoter for a gene that actually would be expressing a cancer cell. So it may not be correcting the gene, but it may be interfering with with basically activating the product of that gene. Those kind of things, and might be kind of just like you've been doing that kind of periodically with some of the areas. But if you think of anything that might be an example, you might kind of say, well, here's a placeholder. We'll talk about it more, but it kind of gives us an idea of early on where there might be opportunities.
Okay. So it's
yeah.
Right now it's mainly concentrated in that last session, but I'll try to work it into the cases as we go along. To also give you little teasers as we go along. Okay. Thank you.
Great.
And this picture here, this picture, we're going to come back to this picture as well because it's important, and also because there's material on this picture that we haven't yet covered. And so parts of this picture are going to be confusing. So what this picture represents is the complete HBB or beta globin gene. This is the gene that ultimately produces the beta globin protein. That protein then forms hemoglobin in red blood cells. And this is the protein, this is the gene that results in the protein that, when it gets mutated, leads to sickle cell disease. So the gene starts here, right at the ACA, okay? And these tan areas correspond to the exons, okay? So you can see the 3 exons, okay? And the other areas here, in small case DNA bases, represent the introns. And the exons ultimately, in a process we haven't covered yet, but which we will, the exons ultimately get transcribed and stitched together and ultimately direct the cell to construct a protein. And again, a little bit ahead of myself, the protein is comprised of a sequence of amino acids, and this picture also shows you the corresponding amino acids for the protein. And that's what the little 3-letter abbreviations are above the DNA sequences. It's the corresponding amino acids in the protein, in the beta globin protein. So the exon starts here and goes to here. And you can see here that immediately upstream of this exon, we have the promoter region. And you have this, what's called cat box, and this so-called tata box, which are where there's a number of known motifs that serve as binding sites for the DNA polymerase, the RNA polymerase. So RNA polymerase knows to bind here and start transcribing the DNA into RNA right here. And it transcribes to here, and another motif, the GT dinucleotide and the AG dinucleotide, are commonly present as signals for the exon-intron or the intron-exon boundaries. And so at the boundary between an exon and an intron, you'll see a GT dinucleotide, and at the boundary between the intron and the next exon, you'll see the AG dinucleotide. this intron is going to be stripped out, and these exons are going to be glued together. The other thing you'll see here is that the exon begins with this ACA, but protein translation won't actually begin until the ATG. Okay, so that's actually where protein translation begins. And to me, one of the things that's very interesting is, I would say there's no doubt in the world, there's no doubt in anyone's mind that the beta globin gene produces the beta globin protein. And there's no doubt in the world, there's no doubt in anyone's mind that a mutation of this particular A, when this A changes to a T, it results in this particular amino acid, a glutamic acid, being changed to a valine. Nobody would doubt that. And so we're all on pretty solid ground. But the fact that the exon starts right here at an A is gleaned through experimental evidence. What they do is they look at the corresponding messenger RNA and they see, oh, well, what's in the messenger RNA? And they can infer back from there where the exon started, but there's conflicting experimental data. And so while I believe there's 100% consensus that this, in fact, is the beta globin gene, and that the beta globin gene, produces the beta globin protein, which then leads to sickle cell disease, there's actually not 100% consensus on where this actual exon begins. And I only say that to try to, I don't want to leave you with what a high school biology student is left with, that all of this stuff we're dealing with is clean and easy. Because by the time we get into next generation sequencing, by the time, getting back to Jim's point that we talked about clinical interventions, and, why is there such reluctance to use CRISPR in actual human clinical trials? I think the more you understand some of the nuances of where these models have some fuzziness in them and where they don't, will help you understand, you can't take next generation sequencing data and just boom, know exactly what's wrong with somebody. And what I want to do as we get into the details of genetic testing is provide you with a little bit better understanding about what are the pros and cons of these tests, what are the strengths and weaknesses of these tests, what's the sensitivity and specificity of these tests. And so to set the stage for that, I tell you now that we don't actually know exactly where the first exon in this gene begins. We're pretty sure it starts here, but some evidence shows that it starts in different places. And what happens is the NIH takes all of this data, they take all the experimental data, and they put together a reference. They put together a reference sequence. And so every time we're looking at DNA or we're talking about DNA variants, what we're really talking about is how does your DNA compare to a reference DNA? And that reference DNA isn't necessarily correct, it isn't necessarily perfect, but it's the best consensus model that the NIH has been able to put together. And so while the reference sequence of this gene might say ACA, that's not the same as saying we know for a fact that's right. It's just we're agreeing that for now we're assuming that that's the reference, so that if you have something different, we at least can unambiguously report back what your variants are. Anyhow, a couple of other things to point out here is you have this sequence, another one of these motifs down at the end of the last exon. This motif here appears to be responsible for triggering the cellular machinery to attach on to the RNA a long string of A's to give stability to the messenger RNA. And then we also see here there's a string of TAA bases, TAA, which terminates the protein chain. So we'll come back to this picture because we haven't really talked about protein synthesis yet. But let me just pause there Any questions about that? Or is it, again, another one of those things where there's just millions of questions and it's a little bit like mind-blowing? To me, when I saw this picture and I realized, that there was nothing structural that signaled where the gene started, it was, I don't know, kind of wondrous in a way, but that's the way it is. So let's return to sickle cell anemia, and before talking about clinical interventions, let's talk about something simple like how would a lab communicate the mutation, okay? So what I want to do is kind of walk you through the process of how we might determine what the mutation is, come up with an unambiguous, publicly recognized identifier for that mutation, and then communicate that variant. There's a number of ways we can communicate the actual mutation. And I'll show you an example of a version 2 genetic test result message, and I'll show you an example of a FIRE message. And maybe next time we'll do a CDA representation. So first thing we want to do, in order to communicate this mutation, we have to be able to name the mutation, okay? And one of the places to go in order to name the mutation is the reference sequence database from the NCBI. And the reference sequences are very important because, again, we can't read DNA and tell you exactly what the DNA is. And that'll become clear to you, especially when we talk about next generation sequencing. We don't know for all of the human chromosomes. We don't know it. We're not 100% sure of the complete sequence of DNA in those chromosomes. There's some areas where we're very sure. There's other areas that are very fuzzy. And so what the NCBI does is it builds a reference sequence. It takes all of the experimental evidence, it does the best it can, and it says, okay, this is a reference sequence of human DNA. And now, when we start naming variants, it's important that we name those variants in relationship to a particular reference sequence. And in HL7, the reference sequence that's used are NCBI reference sequences. So we'll talk about reference sequences a lot. And in this particular case, we're going to search to find the reference sequence for the human hemoglobin, the beta globin gene for the messenger RNA. So let's say we do a search for human HBB, that's the gene identifier, messenger RNA. Okay, so we're searching for that particular messenger RNA. Let's just limit our search to humans. And here we'll find Homo sapiens, hemoglobin subunit beta, messenger RNA. So this is the messenger RNA that's transcribed from the HBB gene. And you can see here it has a reference identifier of NM000518.4. Let's take a look at this. Okay. And let's look at it. We're going to look at it in FASTA format. FASTA format you'll be very comfortable with because FASTA format is the same as what we were just looking at. FASTA format is just simply the sequence. So you see here, here's the ACA, TTT, and here's the ACA, TTT. The FASTA format is just the reference sequence in this particular, in this case, a messenger RNA. Okay. So we're looking at that, and oh, you know what? The other thing I should point out, by the way, counting is a big deal. You'll see when you look at genetics, people talk about zero-based counting systems and one-based counting systems, and this particular mutation is occurring in this A, but how do we number that? And the way that we're going to be numbering it now is to count how far in it is from the start codon, from the start of translation. So this is going to be our number 1, okay? The beginning of protein translation is the signal that's often used to say where exactly a mutation is. And if we count from here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So 20 spaces in from the start is where the mutation is that we're particularly interested in, okay? And we'll see that that is right here. Okay, so knowing that, we can say what the we can now provide a precise identifier for the actual variation in question. Okay. So the syntax used to describe a variant, the HGVS syntax, there's a reference to this in the back of your slides, but you'll see throughout HL7 genomic standards, variations are referred to in HGVS syntax. And in this particular case, we have the reference sequence identifier. So in this particular case, we're talking about messenger RNA 518.4, and we know exactly what that is because we can go back to the NCBI reference sequence database. Sometimes in parentheses, we'll also say what gene it is. And then here, we'll say exactly what mutation it is that we're talking about. And in this case, we're talking about at position 20 in the messenger RNA, an A has been changed to a T. So this is the actual mutation. Now, let's say we didn't know that this was sickle cell disease. You know, you could turn the mutation that you've identified into HGVS syntax, and once you've turned your mutation that you've identified into HGVS syntax, you can now search public databases to see whether or not that mutation has been identified before. Okay. So we can go to the ClinVar database, and this is another NCBI database where you can type in HGVS expressions and you can see whether or not that particular variant has ever been identified before. Okay, so let's say, you know, we found a variant. We can express it in HGVS notation. Okay, now we can search public databases to see whether or not we've ever found this variant. So let's go ahead and do a search of ClinVar. And we find a number of cases where we see that variant. By the way, you know, we'll talk more about this as we go as well. ClinVar is a good source of variant data, but out on the internet today, there is no single comprehensive site of all known variants. So ClinVar is very good. There are private databases that are more comprehensive. There are a number of disease-specific databases. So ClinVar might include, for instance, some of the variants that are responsible for whatever, cystic fibrosis. But if you were to go to a cystic fibrosis database, you might find even more variants in their database. The other thing about ClinVar is that it tries to do the best it can in reporting the clinical significance of a variant, but there's known errors. So you'll see that for some of these variants, it says whether or not they're pathogenic, whether or not they're other. But ClinVar, the pathogenicity of variants isn't always curated by the ClinVar staff. So ClinVar is very good, and it's the main place, I think, to go to look, but it's not the end-all be-all if you're looking for something, particularly if you don't find it here. Now, in this particular case, we're going to look at this link here because this particular ClinVar link only has the variant that we're interested in. These other links have multiple variants. So let's take a look at this variant here.
And Bob, I just had a quick question regarding the previous screen that you were showing. Is most of the association with clinical significance, statistical correlation, or is some of it, coming from, kind of more primary research with cause-effect type? I imagine there's much less data on that, but I'm just wondering kind of what the nature of the clinical significance is. Is it largely statistical correlation or a mixed bag?
It's actually more the submitter asserts it. The people that upload the data assert it. So it's not so much clinical. It's not so much statistics. It's either a curator has reviewed the evidence and asserted that it's pathogenic, or whoever uploaded data to ClinVar that we've done this correlation. We've determined that it's pathogenic. Okay. And you'll see when we look at one of the specific ones, we can take a closer look so you can see.
Okay, great.
There's, as far as I know, there's no algorithmic way to look at the data coming back from ClinVar and say, okay, from a computer perspective, I'll believe it's pathogenic if conditions X, Y, and Z have applied. And so using ClinVar and asserting pathogenicity from an automated tool is a little bit tricky, unless you're already familiar with those diseases, because even though ClinVar is going to report back it's pathogenic, you kind of have to review the evidence as a human in order to decide if you believe it or not.
Makes sense.
By the way, this stuff here is also relevant. So the NCBI produces a reference build. Okay, so there's, we don't know exactly what the human chromosome 11 looks like, but, a few years ago, we took all of the best evidence and we came up with, here's our best guess of a chromosome 11. And we locked it in as a build, and they labeled that build GRCH37. More recently, the build has been updated. These builds get updated it's roughly every 3 to 5 years. So the NCBI will update the builds. And so this is a particular messenger RNA reference sequence, but this is a messenger RNA reference sequence that would have been derived from one of these complete reference builds. And if we go in to look at this one, so now we're looking at variant 15333. Okay, we're looking at variant 15333, and here's the review status. So lots of people have said it's pathogenic. There's no conflicts, so that's good. There's a number of conditions associated with this mutation. For hemoglobin SS disease, you can find more information here in these 3 sites. By the way, the other thing that's kind of interesting is there's no direct link to a SNOMED code from any of this. OMIM sometimes has corresponding SNOMED codes, but in this case, OMIM is going to have the SNOMED code for the sickle cell trait as well as sickle cell disease. And so there's really, today, there's no direct algorithmic approach to go from an HGVS or a ClinVar ID to a SNOMED code. Just something for all of us to think about as a possible service that people might appreciate. So we can look down here. This is the allele ID. So when this particular gene has this particular variant, it also gives it an allele ID. And then when we get down here to clinical significance, we'll see that a number of people have submitted information, and they've all said that it's pathogenic. And based on various criteria that they've submitted. And this disease is not controversial, but you'll see in other diseases, or sorry, not necessarily other diseases, but you'll see in other variations where there's conflicting evidence whether or not a particular whether a particular variant is pathogenic or not. And you'll see there's quite a bit of literature out there about how do you really know. And the American College of Medical Genetics actually has guidelines for how do you even determine if a variant is clinically significant or not. And in fact, it's such an interesting topic that when we talk about next generation sequencing coming up, we're going to discover a new variant, and we're going to be exploring all of these websites because we're going to embark on a journey to determine whether a new variant we've discovered is clinically significant or not. And then to make it even more fun, there's going to be homework. So this is the variant ID. So we can now talk about a ClinVar ID of 15333, and people should know what we're talking about. They should know exactly what mutation we're talking about, etc. And now that we've done that, let's see, I want to go back to that. Okay, now that we know that that's the ID, we can now communicate this particular mutation in an HL7 genetic test result message. And I'm taking this from the September 2016 ballot, and I've highlighted one of the OBXs. Now, I'm not really trying to teach HL7 standards here. If people are looking at these examples and are really struggling with the FIRE examples or the V2 examples or the CEA examples, let me know, and we'll see what we need to do. Hopefully people are familiar enough with the notion of an OBX where I can show you this and basically say, okay, OBX3 tells you is the question, if you will, and OBX5 is the answer. But anyhow, drop me a note or speak up now if people really are lost when we look at these examples. The genetic test result message really is a set of OBXs grouped together in a particular genetic test result panel. And I highlighted this one in bold because if you send this one, then these other ones are redundant, right? You from, and here, right here, 15333, which is the ClinVar ID. The way that they recommend doing it is the code system is ClinVar, the code is the ClinVar ID, and the display name that they recommend using is the HGVS syntax. So given that you know the ClinVar ID, you could just look up what gene is studied, the HBB gene. You could look up what the reference sequence is. You could look up exactly what the DNA change is. You could look up the clinical significance, and you could look up the probable associated phenotype. The allelic state, the fact that it was homozygous in this person, of course, you wouldn't know that from the variant ID. But in this particular, you can also send in the genetic test result message whether in this particular patient you picked it up in both copies of chromosome 11 or whether it was only picked up in one copy. In this case, it would have been heterozygous. So again, it's the simple variant. This is a simple genetic mutation. It's a simple point mutation. Simple point mutations of clinical significance will most often have a ClinVar ID, and the ClinVar ID really then bundles up everything you need to know, including the HGVS syntax, which then includes the reference ID, the gene name, the actual mutation, as well as the clinical significance and the probable associated phenotype. FIRE is pretty similar. In the FIRE resource, which extends the observation resource, they created extensions where these extensions are very similar in meaning to these LOINC codes. And here again, there's a FIRE extension for the observation genetic DNA variant ID, and the value of the DNA variant ID would be the ClinVar ID 15333. And having said that, then the variant name and the gene and the sequence would all be redundant. The allelic state, of course, the fact that it's homozygous would not be redundant. That would also have to be communicated. The FIRE example, a more complete FIRE example, I stuck on the on that same hyperlink. It's just that it's pretty big, and so let's see. I'll have to show you this later. Okay, so you can grab all of these from that examples. Any questions about these examples?
Hey, Bob, how they represent if there is a deletion of a small segment and it's done through some test, so how they represent that information or test results in this kind of message?
Okay, and we will, what we'll do every week is we'll have a case and then we'll correlate the case with the actual informatics processing. For a small deletion, everything's going to look exactly the same. There's going to be a ClinVar ID for a small deletion. Okay, there's going to be a ClinVar ID. There's going to be an HGVS syntax. This is HGVS syntax for a substitution, but there's HGVS syntax for insertions, and there's HGVS syntax for deletions, especially for small insertions and deletions. When we get into larger structural variants, HGVS and ClinVar aren't as useful. We wind up having to use other formalisms. But for small insertions, small deletions, for substitutions, this exact same method would work. There would be, you can figure out what the HGVS syntax is just by looking at the sequence, and then you can plug that in, and you can figure out if there's a ClinVar ID, and then everything else is going to be exactly the same for small insertions and deletions. Not for large ones, but for small ones.
Okay.
Okay. Let's see. Any other questions on this? Okay, finally, this is the closing point. I talk about sickle cell disease, and sickle cell disease is a really, well, I was going to say it's a really good disease to illustrate a point, but there's nothing good about sickle cell disease. Yeah. But one of the things you'll see in sickle cell disease is that it has a very strong genotype-phenotype correlation. Sickle cell disease is always, not always, but let's just say always, sickle cell disease 100% of the time is associated with the 20th base, the A changing to a T, 100% of the time, and 100% of the time that results in the phenotype of sickle cell trait or sickle cell disease. And that makes it, on the one hand, a useful disease to help introduce you to the field of genetics and help introduce you to the flow of information from the genome through the proteome to the phenotype. But on the other hand, most genetic diseases are not anywhere near so simple. Most genetic diseases have nowhere near as strong a correlation between genotype and phenotype. Most genetic diseases, especially in adults, when you're talking about diseases that have a genetic component like hypertension or diabetes, there's multiple genes, each contributing a very tiny amount. There's environmental factors. There's two people that have the exact same allele that differ dramatically in their phenotypic expression of the disease. And a lot of that has to do with the fact that the protein product of a gene interacts with other protein products of other genes, and they form complex networks, and there's tremendous amount of regulatory processes that go in place. And so really, most of the genetic diseases that we're going to be talking about are far more complex than sickle cell disease, and most genetic diseases that we know of are far more complex. But I do think that sickle cell disease helps set the stage for understanding these more complex diseases by kind of illustrating some of the basic principles that we can then say, okay, you know the basic principles, but now we're going to take a look at how it varies. So with that, that's it. Any comments, questions, feedback? Was there good stuff, bad stuff, stuff you'd like to see that wasn't covered? Either talk about it with me now or, drop me a note if you would.
Thanks, Bob. From my own personal perspective, I think this was great. This is, I'm kind of finding this very useful, both reminding me of many years ago when I used to look at this, and kind of I think it's going to be really useful going forward to kind of act as a basis for understanding where pharmacogenomics and testing is going in the future. So this is great.
Thank you, Jim. Oh, by the way, they are trying to treat sickle cell disease. What they do is they do harvest hematopoietic stem cells, then they alter the DNA, and then they try to squirt them back in. They're not using CRISPR. They're using, they're using other genetic engineering techniques. But prior to genetic engineering, really the only cure for sickle cell disease was bone marrow transplant.
Mm-hmm. How persistent are the treatments? Do they revert and then have to be treated multiple times, or?
Well, you have drug therapy. One of the things that drug therapy does is it tries to bump up the production of other hemoglobin molecules. mainly fetal hemoglobin, because that has oxygen carrying capabilities that can kind of offset some of the problems. So the drug treatment, you would have to continue the drug treatment all along the way. If you do genetic engineering, and if you were able to successfully alter the gene and then reintroduce it through stem cells, so far these treatments look to be persistent. You're just cured.
And so
it'd be similar to like a bone marrow transplant. If transplant takes, you're cured.
So they basically would ablate what you have and then replace it?
They could do that. It's, depending on the disease, they can ablate, they can replace, they can try to correct. The techniques are still, there's still quite a bit of techniques, and I don't know. I mean, we'll get into more detail. What's tricky is that while CRISPR is really good at honing in on a particular segment, CRISPR is not so good at doing a very precise single base replacement. Nothing is, and any genetic engineering tool you use might also make changes elsewhere in the genome that you hadn't anticipated. Mm-hmm. And so the tricky part with all of these genetic engineering tools is trying to ensure that you only made the change where you intended to make the change, that the change took, and that you didn't change anywhere else. And in the cases where that's been done, and these hematopoietic bone marrow stem cells have been reintroduced for a variety of diseases, essentially you've had cures.
Cool. That's pretty exciting.
Yeah.
Great. I'm going to go ahead and stop the recording so that I can post this. And by the way, I spoke with a large contingency of the Bay State group this morning, and and I explained that we're kind of doing this initial series, but they said that they'd actually be very interested in attending, because they have a fair amount of clinical people, nurses, pharmacists, and so on that really need primers and want to, they thought this would be a great idea. And I told them that this in the initial series is really an evaluative series, but then later would become probably something we'd offer as a service. And they were very interested. So I'm wondering if this is something that I should extend to them as well.
Well, the more the merrier for me. The more feedback we can get for this go around, the better. I really want to make sure that the informaticists among us are getting the genetic background that they need, so they can feel confident when they start doing genetics informatics work. And if the Bay State folks are able to call in and